Welcome back. This is Rhea, and we are back for our third episode of Neurogeneious. Last time we discussed a wide variety of topics, and now we will be going deeper and deeper into each one. Today, we will be going deeper into the epigenetics of our nervous system and how it exactly links to concepts like neuroplasticity that makes neuroepigenetics a more interesting subject to explore than with other organs or tissues. So, what exactly is neuroplasticity, and why is it a process so important for our brain? We know that for tissues like skin cells, they are constantly regenerating and being replaced, but this doesn't apply for the brain. Neurogenesis, or the formation of new brain cells, occurs obviously early in our development, but it nearly stops to the point where the vast majority of our brain cells can't be replaced, making them very precious. In the face of potential damage to the brain, our body utilizes neuroplasticity to change structures and pathways as a form of adaptation. Neuroplasticity involves the adjustment of many cellular structures, like synapses, essentially junctions between nerve cells where they communicate, and even genes. Synaptic plasticity involves the adjustment of the strength of synapses. Strengthening of the synapses is called long-term potentiation, and the weakening is long-term depression. The process of strengthening or weakening synapses involves either the postsynaptic terminal or the presynaptic terminal, as seen in this diagram. Certain genes in the presynaptic terminal can control the amount of neurotransmitters released. This weakens the strength of a synapse and contributes to the process called synaptic pruning. Other genes on the postsynaptic terminal can adjust the number and types of receptors that bind with these neurotransmitters. They can also affect the channel proteins that allow certain ions to pass through, like calcium or potassium. This last point of mine touches onto another concept of plasticity called homeostatic plasticity, where the excitability of neurons is changed by adjusting the number of protein channels that transmit ions. Now, let's summarize all of this by introducing the genetic aspect of this, specifically referring to phenotypic plasticity in our nervous system. When we refer to phenotypic plasticity, we are discussing how genes or specific genotypes can produce multiple phenotypes in reaction to specific environmental conditions. Phenotypic plasticity is also important for neural stem development, or differentiation, where nerve cells adjust their cellular structures and shapes to enable new functions. There are even identified genes associated with these, which is quite interesting. So now that we've built some good context about the link of plasticity to epigenetics, let's just talk briefly a little bit more about research subjects that still need to be studied pertaining to this. The first thing that we have seen is that there is a huge association of epigenetics and its impact on neuropsychiatric disorders. We can see that because the prefrontal cortex has a prolonged period of development, it is therefore more susceptible to immense changes from the environment. This explains the link between mental health issues and our environments. Lastly, another topic that I don't feel is discussed enough, especially when mentioning neurodegenerative disorders, is the impact of epistatic genes on our nervous system. What exactly are epistatic genes? Well, these are genes that are able to regulate the expression of another gene, even if they are in completely different places on the genome. We consider these to be called inhibiting genes, and we can see that in diseases like Alzheimer's and Parkinson's, there are actually a lot of linked genes as shown here, and even more beyond these diseases in fact. Hopefully, with more people interested in neurogenetics, we can utilize statistical analysis and look deeper into what genetic variations can tell us about the disease progression for these genes, let's say. Anyways, there was a lot of good stuff we went over in this video, and if you feel a little confused on the last bit, don't worry. We'll be going more over it soon, and we'll also hopefully be talking more about sequencing technologies. Thank you so much, and feel free to move on to the fourth lesson.